Welcome to the Sand Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. If you enjoy deep dives into revolutionary research like this one, please do us a huge favor. Take a moment right now to like, subscribe, and share this deep dive with friends and family. It really supports the Sand Science Experience and helps us keep digging into the cutting edge of molecular biology. Today, we are opening up a really remarkable new study. It seems to cut through years of ambiguity to identify the specific molecular trigger for chronic stress in aged neurons. For years, we've known that aging and neurodegenerative diseases, they're characterized by this state of persistent cellular emergency. But figuring out that initial spark, the first domino, really has been, well, frustratingly elusive. This new work points directly at a pretty unexpected culprit. RNA leakage from the cell's powerhouses, the mitochondria.
Yeah, and it's crucial to understand why this matters so much. We're talking about something called the integrated stress response, or the ISR. Think of the ISR as the cell's main emergency defense system. If a cell hits some acute stress, like a virus, or maybe it's temporarily low on nutrients, the ISR basically slams the brakes on general operations. It activates super fast, within minutes, halts protein production, that's translation, and ideally, it returns to normal within about an hour once the threat's gone.
Right. It's like the cellular equivalent of pulling your car over to the side of the road during a sudden blizzard. Saves you from immediate disaster.
Exactly, a perfect analogy. But in aging, and especially in neurodegenerative conditions, that emergency brake just never releases. The ISR gets stuck on, becomes chronically activated. It traps the neuron in this permanent state of stalled function. The cell stops doing essential maintenance, and that leads directly to toxicity and eventually cell death. So the core question this research had to solve was, what's the constant signal keeping this chronic stress response running in aged neurons?
Okay, let's unpack how they actually tackled this. Researchers used a really ingenious model. Human fibroblasts, skin cells basically, transdifferentiated directly into neurons. They called them T-diff neurons. What's so special about this T-diff approach? Why use that?
Well, the T-diff model is pretty powerful because by skipping the usual stem cell phase, these new neurons actually kept key signatures of the original donor's age. So they still had altered methylation patterns, they expressed senescence genes, meaning they function like mature neurons but carried that genuine burden of age right from the start. They had those hallmarks of chronic stress right there in the dish, which is exactly what was needed.
Okay, so once they had these stressed aged neurons, the team systematically targeted the four main components, the kinases, that can trigger the ISR. We're talking about HRI for oxidative stress, PERK for unfolded proteins, GCN2 for amino acid shortage, and PKR, which detects double-stranded RNA or dsRNA.
Right, and they systematically tested inhibitors for three of these, GCN2, PRK, and PKR in those aged T-diff neurons. The thinking was, if the chronic stress was from protein clumping, PK inhibition should work. Or if it was nutrient loss, GCN2 inhibition should help.
But they didn't really work, did they? This is where the story gets really interesting.
No, inhibition of GCN2 and PRK, it yielded pretty minimal results. But when they targeted PKR, the double-stranded RNA sensor, that's when things changed. They saw a sharp, really significant twofold decrease in the number of stress granules.
Oh, twofold. And stress granules are?
Ah, right. Stress granules are basically the physical evidence of that cellular lockdown. They're clumps where RNA and crucial RNA-binding proteins, or RBPs, get trapped when translation stops. So reducing those granules meant the chronic stress was finally easing up. It pointed decisively towards PKR activity, driven by accumulating dsRNA as the persistent signal.
Okay, but you always worry if it's just something weird happening in the lab dish. Did they check this in, like actual human brains?
Absolutely essential step. Yes, they confirmed that dsRNA levels were significantly elevated not only across their different T-diff neuron lines, but critically also in post-mortem human brain tissue. They looked at samples from elderly cohorts, people around 80 years old, and compared them to middle-aged cohorts around 45. The dsRNA pileup isn't just a lab quirk. It seems to be an authentic feature of how our neurons age.
So we know PKR is the alarm bell and dsRNA is the sound ringing it constantly. The next big question is, where's this dsRNA coming from? Is it like a virus or is the cell making it itself?
They use a technique called dsRNA sequencing, very specific, to crack that mystery wide open. And the result was stunning. They found that over 80% of the dsRNA they pulled down in these aged neurons actually came from mitochondrial transcript stamped RNA.
80% from the mitochondria. And they even pinpointed one specific transcript, MT-dsRNA, as the main culprit binding to PKR. Why that one? Why MT-dsRNA?
Yeah, that's the structural detail that makes this so cool. MT-ND6 is unique. It's the only protein-coding gene transcribed in the reverse or opposite direction within the mitochondrial genome. Normally, the mitochondrial genome gets transcribed as these large, forward-running polycistronic RNAs. Because MT-ND6 runs backward, it can easily pair up hybridize with the common forward-running transcripts that overlap its sequence. And boom, that immediately creates a double-stranded RNA structure.
Which PKR then sees as foreign, like a virus, right?
Ugh.
Because dsRNA usually signals invasion.
Precisely. Normally, this MT-dsRNA should stay safely tucked away inside the mitochondria. But the fact that it's activating PKR out in the cytoplasm implies one major thing. The mitochondria are damaged and leaky. Mitochondrial dysfunction looks like the upstream event, the root cause allowing this RNA to escape in the first place.
Okay, how strong was the evidence for that mitochondrial damage? What did they actually see?
It was quite compelling. They ran mitochondrial stress tests on the aged T-diff neurons. For example, they used oligomycin. It's a compound that usually acts on complex 5 to stop proton movement across the membrane. In the aged neurons, they failed to maintain that suppression. It indicates protons were just leaking across the inner mitochondrial membrane, like, as you said, a balloon that's already got holes in it.
And if the membrane's that leaky, does that explain how all that dsRNA gets out?
It certainly seems to. They did further live cell imaging, looking at the mitochondrial membrane potential, CEP. In healthy mitochondria, if you shut down respiration using drugs like antimycin A and rotenone, which target complex 3 and complex 1 of OXPHOS, the membrane potential collapses immediately. But in these aged T-diff neurons, the membrane potential was kind of insensitive to these drugs. It suggests the membrane was already so compromised, running at such a low baseline, it couldn't even react normally to shutting down respiration.
And they actually saw the RNA physically outside the mitochondria.
Yep, they use a technique combining immunofluorescence with fish fluorescence in situ hybridization. They tracked those MT-ND6 transcripts specifically. In the aged neurons, they found clear spots of MT-ND6 localized outside the mitochondrial marker, TOM70, right there in the main cell body, the cytoplasm. It was pretty much smoking gun evidence. The physical RNA was escaping the powerhouse.
Wow. Okay, so that really solidifies the mechanism, doesn't it? Aged neurons develop mitochondrial damage, the membrane gets leaky, MT-ND6 transcripts escape, they form these dsRNA structures which then activate this immune sensor, PKR, triggering chronic stress.
Exactly. And to really nail the causality, they did a crucial experiment. They took young, healthy neurons derived from iPSCs this time and treated them with antimycin A to artificially induce mitochondrial damage. And guess what? That treatment basically replicated the aging phenotype. It caused cytoplasmic MT-ND6 leakage and boosted PKR binding to MT-dsRNA. So damage to the membrane seems sufficient to kick off this whole chronic stress cascade.
Right. So if the problem is PKR driving this chronic stress, the obvious next question therapeutically is, can we stop PKR and maybe reverse some of this damage in the neurons?
That was the really hopeful final part of the study. They treated the aged T-diff neurons with a PKR inhibitor, and the effects were, well, pretty transformative. First, they saw a rapid and significant drop in the main ISR marker, phospho-EIF2 alpha. That confirms the core stress mechanism was being effectively shut down.
The cellular lockdown was finally lifted.
Pretty much, and they confirmed the functional result too. Remember how chronic stress stalls translation and traps key proteins like G3BP1 in stress granules? Using mass spectrometry, they looked at what G3BP1 was interacting with. After PKR inhibition, they saw this striking restoration of G3BP1 interacting with ribosomal components. The protein was basically released from jail and back on the job site.
So the ribosomes could actually start building proteins again, doing what the cell needs.
Precisely. They used Riboseq analysis to confirm this. It showed increased translation efficiency across a whole range of pathways, including restoring the expression of those important RNA-binding proteins themselves. So the pathway seems clear, beautifully worked out, and importantly, targetable. Neuronal aging damages mitochondrium, MT-dsRNA leaks out. PKR gets activated, chronic stress stalls function. Inhibiting PKR stops this cycle and lets the cell get back to work.
Okay, that brings us to our main takeaway from this deep dive then. Damaged mitochondrial leak, mitochondrial dsRNA, kicking off chronic cellular stress in aged neurons, specifically through PKR activation. This identifies a really clear, potentially therapeutically targetable RNA pathway that seems to destabilize neurons and contribute directly to neurodegeneration.
It's a fantastic insight, truly, but it does raise an important sort of follow-up question for future therapeutic development, While hitting PKR with an inhibitor is effective, it stops the alarm bell from ringing. It doesn't actually fix the root cause, which is
Mm,
the damaged, leaky mitochondria and perhaps their failure to properly manage their own RNA in the first place.
right. What does that failure look like? How should they be managing it?
Well, the researchers noted these aged neurons also showed lower levels of proteins like SUV3 and PMPT1. These are part of the mitochondrial machinery whose job is to unwind and degrade excess MT-dsRNA inside the mitochondria before it can cause trouble or leak out. If that internal cleanup crew is failing, the dsRNA builds up and becomes much more likely to escape if the membrane gets leaky. So maybe targeting the mitochondrial quality control system itself, making sure damaged mitochondria get cleared out properly through mitophagy, or maybe boosting those degradation systems like SUB3 that might offer an even deeper, maybe more preventative therapeutic approach, rather than just hitting the stress response after the damage is already happening.
A really fascinating challenge for the next wave of aging research, fixing the power plant itself, not just trying to muffle the alarm it sets off. Thank you for guiding us through this absolutely breakthrough study, and thank you, the listener, for joining us for this deep dive. Don't forget to like and subscribe to support the show and the Sand Science Experience. We'll catch you on the next deep dive.